Welcome back to this next video in which we are discussing the transportation of oxygen and carbon dioxide in the bloodstream. So we've now done carbon dioxide and we're now going to move on to discussing oxygen. So the way I'm going to do this is just like we did when we were discussing carbon dioxide. So when we were discussing carbon dioxide, we started off by discussing the forms that carbon dioxide is present within the arterial blood. So we talked about how carbon dioxide is dissolved in the arterial blood, and we talked about how carbon dioxide is in equilibrium with the bicarbonate ions and the protons within the arterial blood. Then what we moved on to discussing is actually how it's transported around the bloodstream. We're going to do a very similar thing with regards to oxygen. We will start off, however, by discussing it in the venous blood. So we're moving to the venous blood. So we'll discuss oxygen in the venous blood, and then we'll move on to actually discussing its transportation around the bloodstream. Okay, so that's the basic agenda then. I'll pick a nice colour and I'll use this to do our title. So we're now on oxygen. So, oxygen in the venous blood. How is oxygen present within the venous blood? Well, of course, one of the major ways is that it's going to be dissolved in the venous blood, just like carbon dioxide is dissolved in the arterial and venous blood. So this is exactly like for carbon dioxide. The oxygen molecules will be amongst the water molecules. And we want to know then, what is going to be the concentration of oxygen molecules within the venous blood? And this is why people generally don't use concentration, but instead prefer to use partial pressures, because I'm now going to show you a totally different unit for concentration of oxygen molecules compared to the rather sensible unit that we used for carbon dioxide molecules dissolved in the water of the blood. Okay, so the concentration then of oxygen in the venous blood is around 1 milliliter per liter. So this is the units that we use for concentration of oxygen, and you can see that this is very different from the units that we used for carbon dioxide. I'll just go back up here. So we used millimolar for carbon dioxide. Here we are using milliliter per liter. So what this means is if you took a liter of blood and you were to take all of the oxygen molecules out, so the oxygen molecules are amongst the water molecules of the cytoplasm, the red blood cells, and the blood plasma. If you were to separate all of those oxygen molecules dissolved in the fluid away from them and collect them up together, then you would end up with 1 milliliter of the volume of the blood being made up totally of oxygen. Okay, so you can imagine separating out the water and the oxygen and then having a little square left of oxygen of volume 1 milliliter. So this is another way of measuring concentration of a blood gas. And for oxygen, people prefer this unit. For carbon dioxide, people prefer this unit. Now, again, just like Henry's law applied for carbon dioxide, Henry's law will apply for oxygen, and this means that instead of using a concentration of oxygen in the blood, we can instead talk about a partial pressure of oxygen in the venous and arterial blood, and that is in many ways better because everyone can agree on the unit for pressure, and then we quite simply know that the concentration of oxygen in the blood would simply be how much oxygen you get if you expose the blood to that partial pressure of oxygen. So it's in many ways a better way of talking about it. So again, what I'm going to now do is tell you what partial pressure of oxygen this corresponds to. So firstly, I'll start by giving you the Henry's law for oxygen, in particular the solubility of oxygen in the blood at 37 degrees Celsius. So, just like for carbon dioxide then, the concentration of oxygen is going to be equal to the solubility of oxygen, so S subscript O2, times the partial pressure of oxygen in the gas that the fluid is exposed to. And for blood at 37 degrees Celsius, so if we're talking about the fluid of blood and we're talking about the temperature at 37 degrees Celsius, then the solubility of oxygen, and this is where it gets nice. This is a rare nice bit in this discussion. So the nice reason for using this funny unit for concentration of oxygen is because now if we do use that unit, then the solubility of oxygen is actually the same number as the solubility of carbon dioxide. Now, this does not mean that carbon dioxide and oxygen have the same solubilities as one another in blood at 37 degrees Celsius, because we are using different units here, but the number is going to be the same, so that's rather nice. So I'll change colour. So this number is again going to be 0.03, so the same number that we have for the solubility of carbon dioxide, but this time it's in these units, milliliters per litre rather than millimolar, and then of course it's per millimeters of mercury. So for each millimeter of mercury that you have of partial pressure of oxygen, you will get this much concentration of oxygen dissolved in your blood at 37 degrees Celsius. So what this allows us now to do is talk about there being a partial pressure of oxygen in the venous blood. We have this concentration, we can say, okay, what partial pressure of oxygen would I have to expose the blood to in order to get this concentration of oxygen dissolved in there, and therefore we can now speak in terms of partial pressures of the venous blood of oxygen. Okay, so doing the maths then, what is the partial pressure of oxygen in the venous blood? Well, what you'll need to do is divide 1 milliliter per litre by this and find out how many millimeters of mercury you would have to have in order to get this concentration of oxygen. Now think about this. If the partial pressure of oxygen was 100 millimeters of mercury, then the concentration of oxygen that we would end up with is just 100 times this, so it would be 3 milliliters per litre. We've got 1/3 of that, we've got 1 milliliter per litre, so it's going to be a partial pressure 1/3 of 100 millimeters of mercury, which is approximately no, it's not. Get rid of that. It's approximately 33 millimeters of mercury. So 33 millimeters of mercury is approximately the partial pressure of oxygen in the venous blood. Okay, right, so we've now discussed the fact that oxygen is going to be dissolved in the venous blood. We have seen the solubility of oxygen in the blood at 37 degrees Celsius. We've seen that we use these different units for the concentration of oxygen dissolved in there. However, it's nice, therefore, just to talk about the partial pressure of oxygen within the venous blood. Where do I want to go now? I now want to talk about the fact that this is not the only form of oxygen that is in the blood. Of course, most of the oxygen that is going to be within this venous blood is not going to just be dissolved within the water. Instead, it's going to be bound to hemoglobin molecules. So that's where we now need to go. We need to discuss hemoglobin. Okay, so I'll just put a title down here. So hemoglobin is a protein that is present in a huge amount within the red blood cells. So we discussed earlier the fact that carbonic anhydrase is present within the red blood cells. Even more important than the carbonic anhydrase is hemoglobin. And for short, hemoglobin is often just abbreviated down to HB. So I'll go back up to my picture of a red blood cell here, and I'll now draw on a hemoglobin protein. So here, inside the cytoplasm of red blood cells, you have hemoglobin, which is represented by this white square here. Okay, so it's inside the red blood cells, and the red blood cells are absolutely packed of this hemoglobin, and of course, basic biology, it carries oxygen. It's going to have oxygen bound to it. So let's now discuss in more detail the structure of hemoglobin. So we'll start with the protein structure of hemoglobin. So hemoglobin is a tetramer of 4 separate polypeptides that have bound together, and these polypeptides are known as globins. So I'll draw this here. So this square here can represent the entire hemoglobin. The tetramer of globins. We'll see that we have to add more stuff on to actually make hemoglobin. And now I've divided it up into 4 separate proteins or polypeptides that are all bound together to make this tetramer here. Now, what are the names then of these proteins? Well, they are known as globins, and there are 2 different types of globins that you're going to use. One is called alpha globin, and the other is called beta globin. And to make a hemoglobin tetramer, we're going to use 2 alpha globins and 2 beta globins. So let me colour them in separate colours. So this one can be an alpha globin, and this one can be an alpha globin. And indeed, this is the arrangement that they're diagonally opposite each other, the 2 globins of the same type. And then these 2 here will be beta globins. Okay, so this is not yet hemoglobin. So we've got these 2 different types of proteins, alpha globin, beta globin. They are polypeptides. You have genes for them. You can make those polypeptides, great big polymers of amino acids, and you have now bound them together, like so, in this sort of an arrangement. That is not yet hemoglobin. This is just a globin tetramer. To turn it into hemoglobin, you need the heme part of hemoglobin. Oh, and by the way, I should have mentioned for my American viewers that this spelling of hemoglobin is the British English spelling of hemoglobin, the old British English silly spelling of hemoglobin, and I admit that it's the silly spelling of hemoglobin. It's the spelling of hemoglobin that I was taught to use long, long ago, and therefore it's stuck, and I can't easily change, and I just subconsciously write that. However, the more sensible spelling of hemoglobin from the States is like so. You omit the A, and it's a far more sensible word. Okay, so there's the American English spelling of hemoglobin. Right, but back to the structure of hemoglobin, which everyone can agree on. To turn this globin tetramer then into hemoglobin, you need the heme part, okay? So we're going to now add on to the globin tetramer prosthetic heme groups. So the word prosthetic means something that you add on the side of something else. So a prosthetic limb is an artificial limb that's been stuck onto somebody. So a prosthetic heme group is a non-protein chemical structure that is going to be stuck onto each of the globins. So each of the globins in this tetramer is going to have one of these prosthetic heme groups stuck onto it. And these are really important because these are the things that the oxygen molecules actually bind to. And that's how hemoglobin binds oxygen, via its 4 prosthetic groups, one on each of the globin proteins. Okay, right. So let's now discuss heme groups then. So, heme groups. And again, there are 2 spellings of heme. This is the British English spelling of heme, and then the American English spelling of heme is like so. Okay, so we are now going to discuss these prosthetic groups, and I'll write out that name as well. So we refer to these as prosthetic groups that you're going to stick onto the side of the globin polypeptides in the globin tetramer here. Okay, right, so what is a heme group then? A heme group consists of 2 components, so I might just make a little diagram here. To make a heme group, you need 2 components. One of those components is what's called a porphyrin ring, and the other component is that you need a ferrous cation. And a ferrous cation is an iron atom that has lost 2 electrons and therefore has a positive charge of +2. So a divalent positive charge. So Fe2+. That's what I mean by a ferrous cation. If iron loses 3 electrons and becomes Fe3+, that is known as a ferric cation instead. And to make a heme group, you need a ferrous cation rather than a ferric cation. Okay, so we know what a ferrous cation is. Let's now turn our attention to what is a porphyrin ring. Right, now porphyrin rings, there are a lot of different types of porphyrin rings, and they're all based on the structure of a molecule called porphin. So I think the best place to start is with what is porphin. So we'll come up here and we'll discuss what is the molecule called porphin, which is the molecule from which all of the porphyrin rings are derived. So let me draw out the structure of porphin. Porphin is a beautiful ring. It's kind of like in a square shape, and it consists of 4 pentameric rings, which each contain 4 carbons and 1 nitrogen. So I'll just make some space to do this because it's a big chemical structure. So we'll go here, underline the title. Right, so I'll start with the top one up here, and I apologise in advance if this structure ends up being messy. I'll try my best to keep it neat. The other thing I should say before I even start, and I have started, but I'll say it now, I'm going to draw a skeletal structure, and remember, in skeletal chemical structures, you don't show carbon atoms, they're implicitly shown by corners, and you don't show hydrogen atoms coming off carbon atoms. It makes chemical structures look a lot more simple to use skeletal formulae rather than full molecular formulae. Okay, so here is one of these pentameric rings, like so. So the four corners here, 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 and here, those are carbon atoms, and here, of course, is a nitrogen atom. So we're now going to copy this out, but rotated around. So here's another one, rotated round 90 degrees. So here are the four carbons, and there's the nitrogen at the tip. And then another one down here again, rotated round 90 degrees from the previous one, so that the nitrogen head is facing into the center. And then finally, a fourth one over here. Okay, so these are our 4 5-membered rings, each with a single nitrogen and 4 carbons. Now what we're going to do is connect these rings up, and they're each going to be connected up by a single carbon, like so. So each one of these corners is a carbon atom. So this corner is a carbon atom, this corner is a carbon atom, and this carbon atom obviously joins to these two here, and similarly over here. And we'll continue going. So down here, there's that one's connection, and there's the final connection. So now we can see that this is starting to form a nice ring here. And now, to make it a little bit more complicated, we have to add in some double bonds. So the best way to remember this is just to start from the bottom here and work upwards. So this one, we'll put the 2 double bonds there and there, okay? So I've put in 2 double bonds, one is here and one is here. Now we're going to continue on. So we want to now put double bonds into the linkers from this group here, this pentameric ring, to these 2 pentameric rings over here. And there's only one option for where I can put these. I cannot put the double bond here or here because these 2 carbons already have 4 bonds. So if I want to put double bonds on these linkers, they're going to have to go here and here, and indeed that's the way you do it. Right, now we go up to these pentameric rings here, and these 2 are going to be exactly the same, and again we want to try and put in as many double bonds as possible. Now I cannot put them in this ideal format here anymore, because if I put a double bond there, it would mean this carbon would end up having 5 bonds. So instead I'm forced to put them like so, a double bond there and a double bond there, a double bond there, and a double bond there. And this reasoning that I'm going through is my way of remembering this structure. Okay, so we've now added as many double bonds as we can in those rings. So let's now move up to the connections up to this ring up here. So I want to put double bonds into these connections. Again, there is only one option. I cannot put them here and here. I'm going to have to put one here and one here. So I put a double bond there and a double bond there. And then finally, I want to put double bonds onto my final ring. I cannot put them in the ideal starting format, which is down here, because I can't put one there and I can't put one there, because that would mean this carbon and this carbon would have 5 bonds. It also means I can't put a double bond here or a double bond here, so there's only one place I can put a double bond on that final one, which is up there. And this is almost the complete structure of porphyrin. There is one little thing that we need to finish here, and maybe the astute ones among you might have spotted it. This nitrogen and this nitrogen are not happy, they only have 2 bonds, so we need to fix them by putting some hydrogen atoms off those nitrogens. Okay, so that structure that we've now drawn out, and it's got a quite enchanting nature to it, quite hypnotic, beautiful nature to it. It's certainly got a symmetry to it. That structure is porphyrin. Okay, this is porphyrin. And this is the structure from which all the porphyrin rings are derived. So a porphyrin ring, and now I will take the time to write that word out again. So a porphyrin ring or a porphyrin ring, depending on your pronunciation, is a chemical structure that is based on this porphin structure here. So it's something that's very similar to this structure. And I've just realised that's going to get in the way where I've written that. So I'm actually going to get rid of that. We've written it down earlier on anyway, because I need to add things onto this structure in a moment. Right, so going back over here, there are lots of different types of porphyrin rings, and there are therefore lots of different types of hemes. Okay, because a heme, this is the definition, it's a porphyrin ring with a ferrous cation. Okay, we have looked so far at the structure of porphin and there's no ferrous cation yet. That's because porphin is not a heme group. To make a heme group, we need a porphyrin ring and a ferrous cation. There are lots of different porphyrin rings, and therefore we could make lots of different hemes. The type of heme that is present in hemoglobin, that is attached to the globin polypeptides of the globin tetramer, is known as heme B, and this has a very specific porphyrin ring, which I'm going to show you the structure of now. So I'm just going to make some space here and I'll write this down. So the type of heme in hemoglobin is called heme B, and of course if you're American, replace the spelling. And this heme B has a special porphyrin ring, a specific porphyrin ring, I should say, rather than a special porphyrin ring. And this specific porphyrin ring that you use to make heme B is known as protoporphyrin 9. So protoporphyrin, and then the 9 is done in Roman numerals. So I think that is the Roman numeral for 9 there, I and then X. Okay, right. So first thing to do then is to modify this porphin structure that we've got over here into protoporphyrin 9. So I'm going to add modifications on now, and these are the modifications which will turn our porphin structure into protoporphyrin 9. So how am I going to modify the porphin structure then? Well, the way I'm going to do it is I'm going to stick side chains onto the porphin structure. I'm going to have them coming off this carbon, 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 and this carbon. So all of the bottom carbons of the 5-membered rings are going to have side chains coming off them now. Okay, so again, this is quite complicated when you see it for the first time, but I'll show you the logic by which I managed to remember the structure of protoporphyrin 9. So we'll start off up here, and we'll add on the side chains up here. So you have two different side chains, and we'll start off with this one here. So this one here is going to have a propionic acid side chain. So I'll write that down. It's going to be a propionic acid side chain. And propionic acid is the old name for propanoic acid. In chemistry nowadays, they teach you to call this propanoic acid, and I'll put that other name. So the modern name for this is propanoic acid, I think. Propranoic acid? I've forgotten whether it's propranoic acid or propanoic acid. I think it's just propanoic acid. So there's the other name for propionic acid nowadays. So this is just a free carbon carboxylic acid group. So we've got 1 carbon, 2 carbon, 3 carbon, and then on the end you'll have the carboxylic acid group. So I'll put this like so. There's the double bond to the oxygen, and then you could either put the hydroxyl group here, but in reality, under physiological pH, this will have lost its proton, and therefore you'll have an oxygen with a negative charge. You'll have it in the conjugate base form. So this is our propionic acid group, and then you'll have a methyl group as the other side chain. So those are the side chains off this one up here. Now, let's move over to this one here. This one has the mirror image of this one along this line of reflection. So it's going to have a propionic acid side chain coming off this carbon here, like so, and then a methyl group coming off this carbon. So you can see that it is the mirror image of the side chains over here reflected in this line of reflection down here. Now, let's go on to these two. So we'll start off with this one down here. This one again is going to have a methyl group, so there's the methyl group, and then it's going to have a group known as a vinyl group. And a vinyl group means two carbons that have a double bond between them. So I'll draw this like so. So you can see we have two carbons, one carbon here, and they have a double bond in between them. And it might be helpful if I actually just draw out the actual full molecular formula for this. So you'd have two carbons, double bound to one another. Here's the connection to the porphyrin structure here. And then the rest of the bonds would be filled by hydrogens. So here's a hydrogen coming off that carbon, and here's two hydrogens coming off this carbon here. And that is the structure of a vinyl group. Okay? And then this one over here is going to have the same side chains as this one, but this time it's not reflected down the line of reflection. Instead, it's just rotated round 90 degrees. So again, you'll have another vinyl group here and a methyl group. So it's this one, the same side groups as this one, just rotated round by 90 degrees. And this is the structure of protoporphyrin 9. So we're very close now to the actual structure of heme because, well, heme B is the one we're specifically after. This is the one used in hemoglobin. We've got protoporphyrin 9. All we now need to do is put in a ferrous cation. So how do we take a porphyrin ring, specifically protoporphyrin 9, and turn it into a heme group, specifically heme B? Well, the way we do it is we take the hydrogen atoms off these nitrogen atoms, and instead we're going to bind it to a ferrous cation in the middle, and these are going to be covalent bonds. Then what will also happen is these two nitrogens here, they will have interactions with the ferrous cation because they have lone pairs of electrons, and I'll just stick these in. So we'll go over to red now. Okay, so I'm now doing how we're going to convert this from protoporphyrin 9 into the final thing, which is heme B. So the first thing we're going to do is, let me see if I can rub them out. No, I can't anymore because I've written a changed colour since doing the porphyrin structure. The first thing we're going to do is get rid of these, okay? No more hydrogens. And instead, these nitrogens are going to be covalently bound to a ferrous cation here. Okay, so these bonds are covalent bonds, they're going to be shared. Okay, the ferrous cation will give an electron and the nitrogen will give an electron, and they'll form a covalent bond. Okay, this has nothing to do with the positive charge on the ferrous cation, these are just covalent bonds viewing this as an atom. Right, then what's going to happen is you're also going to get less strong bonds, not covalent bonds, but less strong bonds forming between this nitrogen and the ferrous cation. And the reason these will occur is that both of these nitrogens have a lone pair of electrons, and these lone pairs of electrons are a centre of negative charge. Here we have an atom right at the centre with a +2 charge. So these electrons are going to be attracted to that, so they form weaker bonds, and the way we draw these bonds are by dashed lines, like so. So those are the bonds between these two nitrogens and the ferrous cation. So each of the nitrogens of these four rings then has a bond to the ferrous cation at the centre, and this structure that we now have here is the full structure of heme B. Right, so we're now ready to go back and discuss hemoglobin. We have heme B, and by the way, of course, this is a complicated structure. I love these sort of things. I find something very enchanting about them. You know, they're the face of science in my eyes, chemical structures. But I understand that a lot of people don't like chemical structures, so if you just want a simple picture of heme B, what you can draw, and indeed what we will be drawing from now on, is just a box, like so, and then you can put the iron 2+ at the centre, like so. So of course the box is representing the protoporphyrin 9, it's representing all of this bit, and then you've got the ferrous cation shown at the centre. Now, what's key to understand about the heme B is that this ferrous cation can form two more interactions with two other atoms. This porphyrin ring that surrounds it is nicely in one plane, but it can also have an interaction with an atom that's in front of this plane. So I can't really show it with this laser pointer, but in front of the plane where the ferrous cation is, I'll try and show it by drawing in a 3D sense. So it could sort of have another interaction with something that's in front of it, and it could also have an interaction with something that's behind it. So it has two more sites where it's available to interact with other things. And this is the way in which we're going to, one, bind it to the globin polypeptides of the globin tetramer, and two, bind oxygen to it. So one of these is going to be used to bind it to the globin, and the other is going to be used to bind to oxygen molecules. Right, so let's now actually see this. So where is my picture of hemoglobin? So my picture of hemoglobin is up here. I think I'll draw a new picture of... well, no, that's not my picture of hemoglobin, I apologise. This is my picture of the globin tetramer. We haven't turned it into hemoglobin quite yet. We're now going to turn it into hemoglobin. So to turn the globin tetramer, and I'll draw this out again. So here is the 4 globin monomers here. To turn this into a hemoglobin molecule, we're going to bind 4 heme groups, heme B groups specifically, onto each of the globin monomers. So each globin monomer will get 1 heme group. So I might just put the ferrous cation at the centre there. So these are meant to represent the heme B groups, each one of these, and you can see that each of the globin polypeptides has a heme B group attached to it. So, finally, what I want to say is, how do you actually attach one of these heme B groups to a globin polypeptide? Well, it's via histidine residues within the globin polypeptide. So, I want to finish by by showing you a histidine residue and show how the heme B can attach to that histidine residue. So, histidine is an amino acid that is used in many different proteins within the human body, and globin polypeptides will contain histidine residues. So let me show you the structure of a histidine residue and then show how a heme B group can attach to that histidine residue. So I'm drawing the histidine residue as a residue, which means that I'm drawing it as though it's in a polypeptide. So here's the amino group, here's the alpha carbon of the histidine residue, here's the carboxylic acid group, and then the carboxylic acid group is bound to the amino group of the next amino acid along. So now all I need to do is put the R group of the histidine, which is the important bit. So you have a methylene group, and then you have a 5-membered ring with 2 nitrogens and 3 carbons. And I'll draw this in skeletal format. So here is the structure. So you have 1, 2, 3 carbons and 1, 2 nitrogens. And you're going to have some double bonds in this, so you'll have a double bond there and a double bond there, and then to complete it off, we need to put a hydrogen on that nitrogen. So this is the structure now of histidine. And the way in which histidine can attach to a heme B group is via this nitrogen here. Specifically, this nitrogen has a lone pair of electrons, which again is a centre of negative charge, and this centre of negative charge is going to be attracted to the ferrous cation at the centre of a heme B group. So drawing this in, here, this is our heme B group, here is the ferrous cation at the centre, and you can form another one of these interactions that isn't quite a covalent bond, but is certainly an electrostatic attraction there that is enough to hold the heme B attached to the histidine residue between the lone pair of electrons on the nitrogen and the iron cation at the centre. And this is using one of these spare interactions that the iron cation has left. So it can form 6 bonds to 6 different atoms effectively, and we've used 4 here in one plane, and we've now got these 2 left. So this is how you can attach heme B groups onto a globin monomer. Right, so I think we'll have a break there and we'll continue our discussion in the next video. Where we have got to at this point is we now understand fantastically what hemoglobin actually is. It is a tetramer of 4 separate proteins, globins, 2 alpha globins and 2 beta globins, and we know the arrangement of those alpha globins and beta globins here. We then, to turn that globin tetramer into a hemoglobin protein, we need to attach on 4 prosthetic heme B groups. A heme B group consists of a protoporphyrin 9 porphyrin ring with a ferrous cation at the centre, and we have discussed how those are actually attached to the globin monomers via histidine residues. In the next video, we'll talk about how oxygen can bind to the heme groups of the hemoglobin. We'll talk about oxygen saturation curves, and then we'll talk about how oxygen is actually transported around the bloodstream.